Exposure to the pesticide DDT may increase elderly people's risk of Alzheimer's disease. Researchers found
what's at stake is increased risk of a variety of chronic conditions.
Is now playing out in a long-running battle over whether to ban pesticides that harm babies.
Studies show the chemical poses a serious health risk to kids and farm workers.
We're always trying to improve agricultural practices to be more sustainable in the context of our and the environment's health. We believe the next step lies in the plants themselves. If we take a close look at this plant, notice these hairs. These hairs are called trichomes, a plant's natural pesticide found on the surface of many crops, which act as a protective barrier from pests and various elements that are extremely hazardous to commercial farming.
We can advance the outlook of agricultural practices and reduce pesticide usage by genetically manipulating trichome development genes. Using Arabidopsis, we are looking at PLC, a gene notoriously involved in the signaling of trichome development. We previously found that overexpressing PLC will increase trichome expression. In order to verify that the gene responsible for this overexpression is PLC, we're working on knocking out PLC by using gene editing tool CRISPR in the hopes of seeing the opposite effect, a plant with no trichomes.
Last fall, we verified a successful CRISPR knockout of PLC1, one of the multiple PLC sequences found in the genome, as noted by a mutation in the PLC1 gene sequence. However, while a genetic change occurred with the CRISPR knockout, there was no phenotypic or visual change in trichome production or the modified plant, a small obstacle in our research. Along with PLC1, different versions of PLC such as PLC3 and PLC7 work together to ensure the gene's activity is sufficient in the plant. So, knocking out only one version is not enough to see any substantial changes in trichome production.
If we made a hybrid with multiple PLC knockouts, we're more likely to see these changes, which is our next step. Ultimately, our research can be applied to a larger scope of using biotechnology to improve the plants we consume and use in our daily lives.